Hello, my name is Dr. Pierce, and I'm going to be presenting the content on sensory physiology as well as pain physiology in your neuroendocrine course. I am honored and humbled and excited to be able to share this content with you. Please join me as we go through a series of what I call preview videos, which are essentially going to be the exact same content presented in class during lecture, but I provide ahead of time for you in short, more manageable pieces so that you can preview at your leisure in your own timetable. Please don't hesitate to contact me if you have any questions. These are the learning objectives for this content. Please allow me to take an opportunity to explain my approach to learning objectives. Every learning objective has in bold for you, such as this one here I'm underlining, is essentially the task that you need to do as the student to fulfill the rest of the learning objective. In this case, when it comes to explain, you'll be asked to be able to explain or understand the underlying phenomenon that is being taught according to that learning objective. The next part of the learning objective essentially covers the clinical relevance or the reason why this objective is important. And then what follows in the underlined section, such as when I am outlining here, are the actual actions that you will need to be able to do as a student to fulfill the qualifications that are being asked by that learning objective. You will find that my content is broken out by learning objective, and all the information that you need to fulfill these learning objectives are contained within this content. So let's get started with learning objective 1. Learning objective 1 is about the electrophysiological fiber types. We will begin with discussing the naming conventions and classification of peripheral nerves. It is kind of a confusing concept, particularly if this is the first time you have been exposed to this sort of naming system. So let me walk you through it. There are two major classifications that we use to identify a peripheral nerve, at least electrophysiologically. Let me pull up my laser pointer. The first one is up here in the top line. It is saying that we can classify or name any peripheral nerve according to the way that it contributes to compound action potentials. These are essentially the waves of the electrical conduction. And that would be recorded by taking measurements, electrophysiologic measurements, from the entire peripheral nerve. And these are named according to letters, either A, B, or C fibers. Now, a second classification system is based on more of the physical attributes of the peripheral nerve, not necessarily the action potential attributes. And these are based on things like the diameter of the fiber, the thickness of the myelin, and its overall conduction velocity. And these, rather than being named as letters, are named by Roman numerals. They come in classes, class 1, 2, 3, and 4. And while these two schemes of naming conventions are themselves different, they are still related because, for instance, the thickness of the myelin and the overall fiber diameter can contribute to conduction velocity, for instance, which itself is a contribution to overall compound action potential. So the naming conventions can be used simultaneously or distinct, but they are not interchangeable systems. This is, of course, important to understand this now because not only will you be able to communicate and understand the components and expectations of how peripheral nerves should behave, but also you will be able to identify aberrant electrophysiological properties, particularly in the context of various diseases that affect peripheral nerve states. And a classic example of this is in diabetic neuropathy. Now this slide is informational. It is an antecedent to the following slide, which is a very large looking table. What I'm using this here for is to draw your attention that on the next slide, when you see this table, this is a way that I recommend you organize the information about the peripheral nerve classification schemes. You don't need to memorize the entire table, but I did bold some things that you should memorize. And you should also be familiar with the general concepts. How, for instance, the naming conventions are listed from, for example, largest and fastest peripheral nerve conduction to smallest and slowest. And that'll make more sense as soon as you look at the next table. So this is the big table I was referring to, and it probably looks really busy, so let me walk you through what we're looking at. The top half of the table is only referencing information about sensory fiber types, so the afferent fiber types. The bottom half of the table is only referencing information about motor fiber types. Now I understand that this lecture is about sensory neurophysiology, but this particular learning objective does address the understanding and being able to list the group and electrophysiologic naming systems according to fiber type and essentially what they do. I'm not going to go into any detail about what the motor fiber types do because that is covered elsewhere, and I will focus more on the sensory parts, but you do need to know particularly the things that are bolded as I listed in that last slide. So let me talk through the columns now. The first column are the naming systems that are named by letter. These are electrophysiologic classification systems. So contributions of a given fiber type to a compound action potential according to waves. So A waves, B waves, and C waves. You'll note that the A wave fiber types are subcategorized into A alpha, A beta, A delta, and then another A alpha and A gamma for motor fiber types. A alpha motor nerves are not the same as A alpha sensory nerves. That is just referring to similar waves. The next column is referring to the group naming classification system. These are the Roman numerals. Roman numeral 1, subcategorized into 1A and 1B, are A alpha neurons or nerves. Group B are A beta nerves. Group C are A delta nerves, and group 4 are C nerves. You'll note that the group naming system does not apply to motor neurons. Motor nerve, the efferent fibers, they do not utilize this naming convention for the group, the Roman numeral groups. Third column is showing you the size or the diameter of the fibers. You don't need to memorize the specific numbers, 13 to 20, 6 to 12. This is informational, but you do need to know that these are organized from largest to smallest. So A alphas are always the largest, then A betas, A deltas, and Cs are always the smallest fibers. And you'll note that that pattern is mirrored in the motor fibers, but again, you don't need to memorize these numbers. There are two numbers I do want you to memorize, and that is this one in bold, this 80 to 20 meters per second for A alpha fibers. If you think about meters per second, I mean 80 to 120 meters per second, that is essentially covering the length of a football field in a second. That is very fast. And then you'll see that the numbers decrease, and then C fibers, these very small diameter fibers, are relatively pretty slow as far as nerve conduction goes. They're 1/2 to 2 meters per second. Wow, that is pretty fast. I can't run that fast. That is slow when it comes to the nervous system. So again, the numbers I want you to memorize are the 80 to 120, and that is very fast. They decrease in speed down to 1/2 to 2 meters per second, and that is slow. And then information over here in the final column represents the receptor or the function, or essentially what does that fiber type innervate. And these are all bolded. Do know that. Learning objective 2 is about mechanoreceptors. Now let's get into the types of receptors that we use for sensation. In order to do that, we need to differentiate the different types of skin there are, and they're broadly categorized into either hairy skin or non-hairy skin. The hairy skin, such as probably what you are used to thinking about when you think about skin on the back of the hand, on your arm, etc., versus hairless skin is essentially the type of skin on the palm of your hands or the soles of your feet. That's called glabrous skin. And they have slightly different types of mechanoreceptors in those areas, particularly those that are associated with hair and the movement of hair. Another type of category of sensation receptors is how we differentiate them by their ability to adapt to stimuli over time. So when a stimulus persists for several minutes without a change in amplitude or position, the neural response will either diminish at a rate that is very quick, or it would be rapid, or that neural response would diminish kind of progressively and slowly over time. And the difference between those is what we term a slowly adapting neural response or a rapidly adapting neural response. So in this picture, you see the top line shows a stimulus, and that stimulus is essentially nothing at this 0-second point in time. And then at the 0-second mark, a stimulus is applied. And in this case, it doesn't matter how big of a stimulus or anything like that. This is all just very theoretical and something for your mind's eye to picture. But at time second 0 here, a stimulus is applied and that stimulus persists. So that could be, say for instance, taking a pin and pushing the pin against your skin and then holding it there. That would be a stimulus. Now, a slowly adapting neural response is shown in this middle graph, and this is coordinating with the stimulus that is applied at the same time. And this is that kind of hypothetical neural response from a hypothetical slowly adapting receptor. And then if it is slowly adapting, you'll see that there is a big pulse of activity. These are kind of graphic representations of activity neural spikes. You see that there's a pretty big pulse of activity, and then over time the pulse kind of slowly spreads out. And if you were to follow this graph all the way out to what would be its natural conclusion, it would eventually come down to 0 responses over time. A rapidly adapting response, however, means that you get a big pulse at the beginning when that stimulus is applied, but then the pulse essentially diminishes to nothing very, very quick. In this case, within a few milliseconds. So what does this mean for you if you were to translate this into a real-world scenario? So an example of a rapidly adapting response would be you have a stimulus or a sensation applied to the skin, for instance, and after a second or so, even if the stimulus stays there, you quote unquote don't really feel it anymore. And this slide goes into a little bit more detail of the formal definition of a slowly and rapidly adapting receptor. So a receptor that responds to prolonged and constant stimulation is slowly adapting. That means that you get a stimulus applied and you feel it throughout the entire duration while that stimulus is there. A receptor that only responds at the beginning or end of a stimulus is called a rapidly adapting receptor, and that is active only when the intensity of that stimulus suddenly increases or suddenly decreases. And that is what this graphic at the bottom of the page is indicating. This is that kind of hypothetical, this gray line is that sort of hypothetical stimulus application, such as indentation of the skin or somebody pokes you or something like that. And you see that suddenly the stimulus is very low and then something happens, like you get poked on the arm or something like that, and that skin indentation applies, indicating the stimulus activity goes up. And then up above it, let's say you're monitoring neural firing from hypothetical innervation at that area, a rapidly adapting receptor would have a big pulse of activity right at the beginning of that movement. And then the rapidly adapting, that kind of taller bar here in the green, essentially is diminished to nothing. While even though the stimulus is still there, even though that finger is still pushing, say for instance, on an arm or the skin is still indented, the rapidly adapting receptor has not responded anymore. And in fact, that rapidly adapting receptor will not respond until something else changes. In this case, the indentation of the skin withdraws and it becomes the stimulus decreases, the intensity of that stimulus decreases, indicating a change. Whereas the slowly adapting receptor, you see, has a pulsive activity at the beginning. It kind of slowly diminishes over time. You see, the firing rate is spread out here. But then when the change occurs or the loss of indentation, you see that there is again another little pulse of activity from the slowly adapting receptor. And again, it kind of diminishes over time. Now we're going to get into the mechanoreceptors themselves. And there are 4 listed here, but we go into more detail in future slides about the mechanoreceptors responsible for the sense of touch. I like this graphic because it gives you a sense of where they are in relation to each other, for instance, as well as in relation to the epidermal or dermal layers, and then possibly the sorts of structures that they can be associated with. I will specifically draw your attention real quick to the free nerve endings, which are up here in the upper left-hand corner of this graphic. In the graphic, they have actually crossed from the dermis into the deeper layers of the epidermis, and they have these complex branch patterns. Now, these are the very farthest reach of our nerve endings, afferent nerve endings in the skin that provide information about touch, but specifically convey information about pain. We'll get into that later in this content, but you'll notice how complex and very tree-like branching patterns they have, and there are some disease states that really affect these free nerve endings, and we don't know the exact mechanism behind them. But what will happen, say for instance, in diabetic neuropathy, is these free nerve endings will essentially kind of die off, and then they retract farther deeper into the dermal layers. When they do something called crossing the epidermal-dermal border, that means the nerve endings essentially kind of shrink back and get pulled back into the dermis, such that they're no longer innervating the epidermal layers. Now, that has a lot of consequences for a patient who have a retraction of these free nerve endings. If you suddenly lose in a patch of skin or somewhere, say for instance, the bottom of your foot, free nerve endings in the epidermis, that means that you can have damage to the epidermis and not really feel the pain associated with it. And in fact, that is one of the reasons why someone with a diabetic ulcer, for instance, on their foot, doesn't necessarily feel the pain from that ulcer and know it's even developing, because those free nerve endings have retracted so far, they've essentially shrunk back or died so far that they don't work anymore. The person doesn't feel pain in that area. So you can imagine, very important concept. And we'll get more into that later in this lecture. So I like tables. I think tables are a great way to organize information as well as test your own memory and test your own understanding of content. This is a table that breaks out the mechanoreceptors, particularly those that I will be holding you responsible to. The ones I will be holding you responsible to are listed in the learning objective. So if you are wondering, will Dr. Pierce test on this? Um, always look back at your learning objective for an answer, or you can feel free to ask me as well. But this table has your mechanoreceptors listed out here on the left, and then some muscle mechanoreceptors that I'm not going to go into a lot of detail on. Those are the muscle spindles and Golgi tendon organs. Dr. Carius will be teaching that content. As far as the receptor types, they are found in this middle column. There's information in there about the sort of threshold that they have, if they're quickly or rapidly adapting, and what sort of skin they're found in. And then, very important, and you definitely need to know these, what type of sensation is associated with that mechanoreceptor. And if you think about it, try to assimilate in your mind the function of that receptor. So for instance, vibration, for instance, with where it is, how deep in the skin is it, if it's rapidly adapting, etc. So for instance, vibration is a sort of sense that you really pick up on vibration when it starts and when it stops. And if you hold something that vibrates against the skin for a period of time and without changing the intensity or the duration or anything, just kind of keep it steady, that sense of vibration really kind of wears off over time. You don't really feel it as much. Well, that's because it's really working on those rapidly adapting Pacinian corpuscles. And you saw in the previous slide that Pacinian corpuscles are found very deep within the deepest layers of the skin and the dermal layers themselves. And so it takes quite a bit of indentation of the skin, as would occur from vibration, that kind of powerful mechano force, to stimulate a Pacinian corpuscle. So that's my recommendation to you, is sort of assimilate this information in your mind, see if it makes sense. Why would a mechanoreceptor that's slowly adapting transduce information about pressure, for instance? So think through those things. If you have any questions, let me know.